So biogenesis is spontaneous generation. So this is when living things are arising from non-living things. Um, and there was a scientist named Reddy, and he disproved a belief that flies were spontaneously generated from rotting meat. So before his experiment, people thought that rotting meat created flies, not attractive flies, but actually that flies came from meat. Okay, because flies always seem to be around rotting meat. So what he did is he had this experiment, and you can kind of see it in this picture here. He had 3 different jars. He had a jar that was open and meat in it, and then he had a jar. So this was like his first set, and then he had some jars that were like completely sealed, where nothing could get on them, and then he and no smell could get out, nothing like that. And then he had jars covered with like cloth netting. And then he left them there and let the meat rot. On the open jars, they were completely covered with flies at the end. You can see there's a lot of flies here. On the sealed jars, the meat rotted just the same, but there were no flies on the meat, not a one, because they couldn't get in there. And then on the jars covered with netting, where you could smell it and that kind of thing, but you couldn't actually, the flies couldn't get to the meat. There were a whole bunch of like fly eggs on the top. And so this is pretty simple experiment, honestly, but it actually kind of disproves the fact that flies come from rotting meat. Because if flies came from rotting meat, the tightly sealed jar would have some flies inside as well. And then the jars covered with cloth netting would have flies inside as well, because the flies were coming from the meat, which is not what happens. So this thing that all living things come from other living things is called biogenesis. Something has to be alive. So like rotting meat cannot make living things come from it. Living things can't come from rocks. They have to come from something else that is already living. So that's kind of where that came from. There's some other experiments that were kind of interesting. Spallanzani had an experiment where he took meat broth and he boiled it to make sure there was no like living organisms and things like that in there. And then he closed one flask. So he sealed it somehow. And then so this is kind of what Spallanzani's looked like. Right, here's the flask. So he had some meat broth in there that had been boiled, and then he basically like put a lid on one of them and then left the other one open. The closed flask didn't grow any bacteria, but the open one had all sorts of bacteria in it. Pasteur, he did an experiment. He used a curved neck flask. That's what you're seeing in these pictures. Basically, that prevents microorganisms from entering. If you put some water right here, then the microorganisms can't get to the rest of the flask. He did that instead of sealing it. So he boiled it and he let it sit. When it was sealed, there was nothing in there. Then he boiled it again, and then he took he kind of took the curved neck off and let it sit, and then there was bacteria and things in there. So that was, again, showing that it had to be exposed to the air in order for microorganisms to appear, and then it just didn't automatically appear. So that's kind of how they started finding out that organisms came from other organisms. In general, we think that Earth is about is over 4 billion years old. So that's billion, right? That's very, very old. And basically what we do is we use something called radiometric dating. And we measure the quantity of a particular isotope to determine the object's age, and it's usually carbon-14. And so basically an isotope is an element that has different numbers of neutrons. So you can have carbon 12 or carbon 14. And then the half-life is how long it takes that sample to decay. And the half-life of different elements is known. And so if you know how much you started with, for example, let's say that a sample starts with 10% carbon 14, right? And then it turns in carbon 14 turns into carbon 12. If you've got 95% carbon 12 and only 5% carbon 14, you know that 5% of that carbon 14 is decayed, or half of it. Okay, so if the half-life of carbon 14 is 10,000 years and you've lost half of it in that time, then you know that sample is 10,000 years old, give or take. So that's kind of how they figure that out. And so they basically can take the age of things like rocks or fossils and things like that, and that's how they get like the estimated age of those sorts of things. So there's two theories as to how the very first organic molecules were formed. Okay, because when Earth was first formed, there was no life on Earth, right? Somehow the life had to appear. They think that the first theory says that organic molecules were formed from a chemical reaction involving ammonia, hydrogen gas, water vapor, and methane. And in 1953, Miller and Urey were able to produce organic compounds from inorganic compounds. So that made them think, okay, maybe this theory is what's going on. And then another theory is that organic molecules arrived on Earth from like a meteorite. And in 1970, they found organic compounds on a meteorite. So that made people think, well, maybe this is how it happened. It might be a little bit of both, we're not entirely certain, but these are the ideas as to how we think that the first organic compounds appeared. Okay. Now, there's some questions about how heredity works. For example, DNA needs to replicate. Why does DNA need RNA? Why can't DNA just replicate itself, right? Why do you need that RNA step? And there's these things called ribozymes, and they're basically RNA molecules that act as a catalyst and promote specific reactions. And they can act as catalysts for their own replication. So they're thinking that maybe RNA came first and then DNA. And so that's why DNA needs RNA, because RNA was first. There's also some viruses that only have RNA, no DNA. Now, some of the first cells did something that's called chemosynthesis. So they actually made energy from inorganic compounds. So instead of like eating things, they actually were able to make energy from things like sulfur and things like that. And there's actually some organisms and bacteria at the very bottom of the ocean. I know you're told, like, when you're growing up, that all food webs begin with the sun. Well, there's actually some food webs in the very, very deep ocean that are at hydrothermal vents. So there are these places underwater where they're like this, and then like inorganic compounds kind of like seep out of them. And they're so deep underwater that there's no way that sun can penetrate to this, like miles underwater, that there's no way that sun can penetrate to these ecosystems. And so the organisms that live here, there's like little worms and bacteria. There are some crabs. These organisms, they all depend on this this inorganic material coming out of these hydrothermal vents. So this is actually an ecosystem that does not need the sun. So that's kind of, a big deal when they found those out. Cyanobacteria are a different thing. They are photosynthetic and they're unicellular prokaryotes. So they're bacteria, but they also do photosynthesis. And this is one of them, an example of them is Limbia. That's this stuff. I used to have to go around the water, different areas in Florida, and scuba dive and collect this stuff. Sometimes they call it witch's hair. because it's so yucky looking. It's really gross. And so basically, these are prokaryotes, so they don't have a nucleus, but they can do photosynthesis. And so if they're doing photosynthesis, right, they are taking up CO2 and they are releasing oxygen. Okay, and so if you have a lot of these organisms, eventually, right, after a long, long, long time, like billions of years, the oxygen concentration in the atmosphere will increase, which will allow the ozone layer to form, which then can protect us from the sun's radiation. So that makes the environment a little bit more hospitable, but it also makes it to where organisms that breathe oxygen could maybe survive. So the first eukaryotes, these are critters that have a nucleus, um, and they have also little like they call them organelles. They're kind of like organs, but they're not really because they're single cells. So they call them organelles, and they are eukaryotes that evolved from a symbiosis. They think between primitive eukaryotes and the prokaryotes they partnered with. So, for example, chloroplasts. They think like, for example, plant cells, the first plant cells were maybe a symbiosis between a photosynthetic prokaryote and a larger prokaryote. So they kind of live together, kind of like how algae cells called zooxanthellae live inside a coral. That's a symbiosis in the coral cells. And then mitochondria, they think, was again, a prokaryote that had some characteristics of mitochondria, and they formed a symbiosis with some other prokaryote. And if you remember from your study of cells, mitochondria actually have their own DNA. So your DNA is actually like 50% mom and 50% dad. Well, your mitochondria has DNA and it's mom only. So your mitochondria is exactly the same as your mother's, and your dad's mitochondria is exactly the same as his mother's. So your mitochondria isn't anything like your dad's mitochondria. They're different. Um, and so they think because mitochondria have their own DNA like that, they think that maybe they used to be their own critter.